All right, ladies and gentlemen, welcome back. So today we're going to start getting into the different types of transport that your cells participate in based on the molecules that we're moving and the chemistry of that phospholipid bilayer. So again, remember that your membrane structure results in selective permeability. The fact that those fatty acid tails in the center are hydrophobic, nonpolar, regulates what is allowed to pass through and what's not based on what those fatty acid tails are capable of interacting with. And then we can take into consideration protein channels that can make basically hydrophilic tunnels to allow other things to get through. So a cell has to be able to exchange materials with its surroundings, period. It needs to get in nutrients and be able to get out waste products. But we want to be able to control how much goes in, how much goes out, and when. And so the structure of that plasma membrane is going to allow for those cells to be able to control that flow. So keep in mind that hydrophobic materials, right, lipid-soluble materials, hydrocarbon type materials, anything that is capable of interacting with those fatty acid tails is going to pass through that membrane rapidly, especially if it's small stuff. All right, so anything that's hydrophobic and small is going to be able to pass directly through those fatty acid tails without any issue because it can interact with those fatty acid tails. The polar molecules, on the other hand, or anything that's ionic, I should also add there, so make a note for yourself there. Anything that's polar or ionic, aka anything that is hydrophilic, isn't going to cross that membrane rapidly. Assuming that it's small enough to get past that membrane, things like small sugars, sodium ions, calcium ions, that kind of thing, they're going to need transport proteins to allow for the passage of those materials across the membrane. Those transport proteins are also specific to the substances that they move. So some of those proteins can just be channel proteins, meaning that they literally are exactly what they sound like. They are just pores that are opened on either side of the membrane, and they will allow things that are hydrophilic to pass through them. So that means that that internal amino acid chamber has a bunch of hydrophilic amino acids there so that they can interact with those hydrophilic materials. And then out here are the hydrophobic amino acids interacting with the fatty acid tails. So they can literally just be tunnels. They can also be carrier proteins, which is kind of what this guy is showing. So it's typically opened on one side of the membrane. It will attach a molecule when it's present, change its shape, and shuttle it to the opposite side. Even your water needs proteins to get through, right? Water is by definition polar and hydrophilic. So it's not going to be able to interact with those fatty acid tails, that hydrophobic region. So instead, they have channel proteins, specifically called aquaporins, that facilitate the passage of water through them so that we can allow that water exchange to occur without having to fight the fatty acid tails in the middle. So depending on what we're moving and in what direction we're moving it, then determines whether or not energy is necessary. So if we're moving something passively, passive transport, this is moving substances across that membrane, polar, nonpolar, ionic, it doesn't really matter, but we're not going to need any energy investment to do it. AKA, there's no ATP involved here. So all sorts of things, carbon dioxide, water, oxygen, all of these things diffuse across the plasma membrane really easily. We don't need energy to move them. Might we need a protein to do it? Sure. But I don't need energy to make those proteins work if they're involved. Otherwise, they just pass straight through the plasma membrane because they can interact with those fatty acid tails. The carbon dioxide, the oxygen, for example, just goes straight across the membrane, straight through those fatty acid tails. They don't have a problem interacting with them. The water, as we just talked about, will use the aquaporins, so they'll need the protein, but I don't need any ATP to make that protein work. So we're talking about diffusion here. And when we specifically talk about diffusion of water through those aquaporins, then we can talk about osmosis. So when we talk about passive transport, we talk simple diffusion, we talk osmosis, and then we talk facilitated diffusion. So let's kind of walk ourselves through those three types of passive transport. Okay, so let's start with simple diffusion. And diffusion, by the way, doesn't even require a membrane to be in place. We can have diffusion of scents happen in a locker room when you spray a bunch of perfume and you can smell it eventually across the room. Um, Candles, that kind of thing, or putting some food coloring into a beaker of water and that dye diffuses. That's all diffusion. There's no membrane involved. Now, when we talk about diffusion in cells, naturally the membrane now becomes a selectively permeable barrier for those molecules. So when we talk diffusion, it's just the tendency for molecules of any substance to spread out evenly in the space that it's provided. What does that mean? That means that wherever that initial drop of dye occurs, or that initial spray of perfume happens, or the initial deposit of some solute occurs in an environment around a cell, those molecules are going to go from where they are highly concentrated, right? We call concentration gradients. So where they are highly concentrated to where they are lower in concentration. So there's an overall net movement, and that word net is really important. There's an overall net movement of particles from an area of high concentration to low concentration. That being said, if I go and I look at any individual molecule, that molecule may be moving this direction, it may be moving this direction. Because all it really comes down to is having all of those molecules really tightly packed together that they're bouncing off of each other. Um, and so what they'll do is they will think about your simple laws of physics, right? Every action has an equal and opposite reaction. They bounce off of each other, they spread each other out, and eventually they get so far dispersed that they're no longer hitting each other. And that's when we start talking about equilibrium, right? When they have balanced each other out to the point where they are far enough apart that they're rarely hitting each other and throwing things in other directions. All right, but while that concentration is really high, they could be bouncing all over the place. Again, kind of think of packing the cafeteria full of students, right? The general net movement is moving out of the doors and spreading out into the building. But any individual one person in that cluster, that in that crowd as you're trying to move through the door, may be getting pushed or shoved in opposite directions that may not necessarily be towards the door itself. And that's exactly what's happening at a particulate level, at a molecule level here. Once we reach that dynamic equilibrium, it's dynamic, meaning that those molecules are still moving, but the net movement of molecules is no longer occurring. We've reached equilibrium, so I have the same number of particles on one side of the membrane as I do on the other. So let's just say there were 20 dye particles here. By the time I get here, that would mean I'd have 10 on this side and 10 on that side. And it's not that those molecules aren't moving back and forth, they are, but they will continue to maintain 10 and 10 on either side.